So, we've seen this before, right? This central dogma idea of going from DNA to messenger RNA, and then from our messenger RNA, so of course that was transcription, right? Then from our messenger RNA to our protein is the second half of our story. So this is what we want to focus on now. As we read in that little introduction to that activity, right, this is called translation because we're going into a very different language. Nucleic acids, very, very different language than amino acids, right? And so we're translating from one to the other, and it's going to be that ribosome that's going to help us to do that. That ribosome is going to be like a factory building more copies of these proteins, right? So that's what gets us into 10.3, which you read for today, 10.4, which you read for tomorrow. 10.3 was a little bit more of an overview, kind of reminded you more about the codes and codons more than anything else, I think, right? And 10.4 gets a little bit more technical into the machinery, and it's going to talk a little bit more about this ribosome and those three sites I talked about, A, P, and E. It's going to talk about this transfer RNA and how transfer RNA actually is able to carry the amino acids over. And those points, 10.4 especially, are going to be important for you to be able to kind of reenact that tomorrow on our messenger RNA strands. So if we look at this code, right, so we got our DNA. Notice it starts with TAC, right? That code should kind of jump out to you now that we talked about that starter sequence. This TAG is going to be what for RNA? AUG, right? And again, we'll review ourselves on this little code here, but AUG is going to give you methionine. So basically this DNA code, we're just kind of skipping the promoter part, right? Which is pretty common in terms of simplifying things. We just kind of, well, like we did sophomore year, right? We just kind of said, oh, no, we won't worry about that promoter stuff. We'll just kind of jump into the transcript right here. And so we kind of move on here. We go from our DNA to our messenger RNA. And so here we got our AUG, right? And then we go down, we start to translate. Can someone, I don't think it's probably too complicated, but it's been a few years for some of you, right? Using this code, right? This box, someone left theirs back there, but make sure you hang on to this. And so we've got AUG, there's different boxes, there's different wheels. I don't know which one you use fresh or sophomore year, but on the left side we go A, right? This is our first base. Our second base is U, so we're in the first column. Our third base is G, so we're on that bottom row of the fourth column, and that brings us over to that highlighted box where it says methionine or start. And again, that's how we should always be starting our translation. We should always end up with methionine. Now we'll find out later that not all proteins actually have a methionine at the start, and so it always will have methionine, but sometimes it gets modified before it's done and it chops that methionine back up. Now, methionine doesn't always have to just be at the start either. It's still possible to get an AUG code elsewhere, right, and to bring a methionine kind of into that protein somewhere else, right? And so that's the general idea. Again, kind of the simplified form about how this messenger RNA is going to lead to an amino acid sequence. And that's kind of the extent of our focus that we had in your first year of biology, right? We didn't get into too much of the mechanism stuff. We were just curious or concerned with the fact that you could use a chart like this, or sometimes it's in a wheel form, and come away with these amino acids. Now, that's ultimately what you're going to do tomorrow, but we want to simulate it a little bit more accurately. Now, the idea of how you can code for 20 amino acids with only 4 nucleotide bases, right? And that's the idea of how we discovered that there are these things called codons, right? These things called codons. And so, I'm going to kind of fast forward through these things a second. You can see that we're going to start to divide things up into groups of 3. And it was Crick, of Watson and Crick, that came up with this idea of using this three code sequence. And the reason why is, and it's kind of the same reason why a lot of people were skeptical that DNA was the genetic material, because you look at it and you say, oh, you have 4 choices. Well, if your code is an A, you can't have an A bring in 5 different amino acids, right? And you can't have a C bring in a different 5 amino acids, because how would it know which one to bring in? If you had codes of 2, how many options would you have? You said it read it in codes of 2. So AA would be a code, AC would be a code, CG would be a code. Maybe 4 to the second, 4 different bases with 2 options, 16, right? Again, won't quite get you there, right? And so you're like, okay, so I have 16 options. I still have 20 amino acids, which would mean AA might code for 2 different amino acids. And again, that would be a problem, right? If the ribosome reads AA and it says, I wonder if I'm supposed to bring in glycine or valine, you're going to have an issue there. So good, not good enough. But if we go to groups of 3, right? 4 to the 3rd, 64, right? Now we have more than enough to cover our 16 bases. And so if we look in here, we can see there's 64 different codes going on here. But it also explains why, well, we have 64 codes for only 20 amino acids. And actually 3 of those codes are stop codons, which means we actually have 61 codes for the 20 amino acids. Well, that's why some of them are used more than once, which your book called redundancy. And so you look at leucine. Leucine, CUU, CUC, CUA, CUG, UUA, and UUG. All of those codes are going to bring in leucine. Other amino acids only have 1 code. Okay? And so it was Crick that determined the idea that it has to be codes of 3 in order to be able to have this, to be able to have each amino acid have a unique code, right? But it was someone else who actually discovered which ones actually pair up. So he came up with the idea. Another group of scientists figured out that AUG is going to pair up with methionine, and that CGU is going to pair up with arginine, GUA is valine, on and on and on, all the way down to GCC being alanine. And so we get this group of scientists. Again, Crick was the one that kind of determined this codon system, but what didn't actually work out which ones matched which ones. But it was these 2 guys, Nuremberg and Corana, I think is how you say it, that actually went through and they kind of figured out which ones matched. Now this is similar to what we did the other day. You look at this and it doesn't make a whole lot of sense right off the bat. And then you start to see, wait a minute, what if I broke it down into 3s, right? And so if we do a little color coding there, it starts to stand out a little bit easier. And why did the red bat eat the fat rat, right? Reading things in groups of 3. Now, in terms of trying to figure out, now the next step was to say, okay, so what does CCC code for? What does UAG code for? And it were these 2 guys that discovered it. He discovered 47 of the codes, right? So he discovered CCC was this and ACG was this. He discovered, and he did 17 of them. What does that add up to be? 60. Oh yeah, that's actually, okay, 64. Sorry, I was thinking that they didn't do the stop codons. I was thinking 61. But, and again, 3 of those codes come out to be determined that, wait a minute, this doesn't actually bring an amino acid. This stops the translation code. That must be a stop codon, right? And again, you can see that in the red here, they received a Nobel Prize for this work, figuring out basically how to decode the system. And the fact that, hey, this system works in humans. It's the same system in a rat. It's the same system in a plant. It's the same system in, and you go through all these different living organisms. It's the same 4 codes. It's the same codon system. It's the same idea of bringing in or pairing up one of the codes with a specific amino acid. And so these guys determined the messenger RNA/amino acid match. Basically, they discovered that they could actually use kind of fake RNA made up or fabricated messenger RNA, which was a whole lot easier. They had them to different test tubes that contained ribosomes, that contained transfer RNAs, that contained amino acids. And by being able to add these kind of fabricated messenger RNAs, they were able to make their own sequences and see which amino acids ended up being bound to it. For example, they would create a UUU. Notice they got several U's in there, right? And so they have UUUUUU, and what do they find all attached to that thing? They find phenylalanine, right? Pretty simple experiment, right? But pretty good idea. And then they would do a different combination and find out, oh, well, hey, look, we always get this amino acid bound to that. And so that led to this idea of the genetic code, right? The codons that now we just have this handy-dandy little sheet to tell us which ones are going to be matched up. So just a couple pictures of them looking happy and such. So here's our code that we were just looking at as well. We can see our methionine start code right there. And so again, this is the code for all of life, right? You had a couple questions that said, you know, what does this code being universal imply, right? Well, It implies that it's the code for all life. It could be an argument for kind of a common ancestry thing. Again, please be critical when you read things about that and don't just read your book and accept things, right? You got to think about, what you think about that. Are there other explanations? It's obviously your textbook's not going to lay out, well, maybe having everything have the same code means that it was created by the same creator. Right? Does that seem logical too? It's logical to me, right? So anyways, it is important to point out though that all things do have the same code, right? And your book spent a little paragraph and it actually gets into chapter 13 with it, but since everything has the same code, we can actually kind of mix and match things, right? Which is why you can take a human gene for insulin and you can splice it into a bacteria. And that bacteria can still read that gene and still transcribe it, still translate it, and actually make human insulin in the bacteria, which then you can extract and give back to a human, right? A pretty amazing thing when you think about it, but since it's just the exact same code, it knows how to do it already, right? It has the machinery to read it. The code is redundant, which means that several codons for each amino acid. And so it's redundant. And so your book talks about that it's good that it's redundant. I think not ambiguous is how your book phrased it. Ambiguous would mean that you would have one code, and it would code for several different amino acids. That would be the problem, because in different situations you get a different amino acid there, right? But it said it doesn't matter that it's redundant. It doesn't matter that there's several codes, because any of those codes are still always going to bring the same amino acid. Now, how can you explain this right here, that leucine has 4 different things, 4 different possible codes just right here, and it's this idea termed the wobble. Notice, CU, 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 CU. All 4 bases are the same, or all 4 of them have the same 2 bases to start. The third base is considered a wobble, and really what that means is it's a little bit different on how it binds. It doesn't have to bind as tightly to that third one, and so it really doesn't matter. Basically, what it also means is that there's not 61 different transfer RNAs floating around, right? Hopefully you caught that in your book. It says there's about 2/3 of that. There's not 61 transfer RNAs because you could potentially have one transfer RNA that would be able to match up to all 4 of these codons. Because of that third base, when it pairs up, it doesn't have to form as strong of a base. There's a little wobble in there. There's a little flex room in there for it to still bond to it. So the same amino acid, or the same transfer RNA bringing the same amino acid, could bind to all 4 of those codons. Now you have the start codon, which is your AUG, and you also have 3 stop codons, and those we can see over here, UAA, UAG, UGA, all kind of similar there, are all going to stop this translation process, which means whatever you've made in terms of amino acids, once you hit that stop codon, the ribosome is going to break apart, the messenger RNA transcript is going to get released, maybe to find another ribosome, and that polypeptide chain that you just made, sequence of amino acids, also going to get released, and now it's going to do its folding thing, right? Take on the secondary, the tertiary structures, etc., and be functional. And so, basically, those only do stops, but we don't have a specific start one in terms of it does more than just start. It actually does bring in amino acid, whereas the stop ones do not bring in amino acid. It's just a pure stop command. So why is the wobble good? Well, probably the easiest explanation is it allows you some flexibility. For one, it allows you to not have to have 61 transfer RNAs to account for the 61 codons, right? And so it also, well, basically just that reduces the amount of raw materials that need to be produced in your cells as well. It's probably a good start. So how do we match these codons to the amino acids? Well, we've already kind of seen this diagram, right? So we take our transcript, or our template, right? Now remember, this whole 5' to 3' thing, you got to be a little careful with, because when we say that, we said back in chapter 9 that replication happened from the 5' to the 3', we were talking about 5' to 3' on the new piece that was being made, right? Same idea here. Transcription is going to happen from the 5' to the 3', but again, we're talking about, that's the actual transcript, the messenger RNA, right? So our DNA is actually 3' to 5'. Our messenger RNA was built this direction, 5' to 3', because our RNA polymerase has the same problem that DNA polymerase has in terms of it needs to have that 3'. It can only add to 3', right? So if this is my 5', I have a 3', 3', 3', right, and it adds in going this direction. That being said, RNA polymerase is a little bit more efficient than DNA polymerase because it does not need a primer to start it. So it can start it itself, but it can only build in that 5' to 3' direction. Then, of course, we break things up into groups of 3, and then we get this idea of a transfer RNA, which you'll read about for tomorrow. Our transfer RNA, if this AUG is a codon, what's it on the transfer RNA again? We'll read about for tomorrow. I thought maybe we do something. Anticodon, right? Codon, anticodon. They are complementary to each other, right? It's all RNA, so A's with U's, C's with G's. There's no T's in either of those, right? And this UAC transfer RNA is always going to be linked with methionine, right? That's the one that's going to carry. And when the amino acid is on the transfer RNA, your book calls it it's charged. That transfer RNA is charged. It's carrying the amino acid it's meant to carry. And so it comes up, and the ribosome can actually do this for us, but it base pairs with it, and it brings the correct amino acid to the party. Then, the next, now we have CGU, so GCA, complementary transfer RNA, which happens to be carrying arginine, brings arginine to the party. And then GUA is going to bring CAU with valine. And now we're going to start to form, what's the bonds between amino acids? They are covalent with the name. Peptide bond. And so we're going to form peptide bonds between them. Probably not quite something you remember from chapter 3, but proteins, polypeptide chain, are built from the N terminus to the C terminus. If you remember amino acids, they have an amino group on one side, that's the N terminus, and they have a carboxyl group on the other side, that's the C terminus. So that's N, C, N, C, N, C, right? So from the N terminus, amino group point out that way, to the C terminus, the carboxyl group going that way. And so it's being built like that with an anticodon.